Started?
Yes.
Okay. Or do you have any announcements?
Not at this point, maybe at the end I'll mention a couple of things.
Okay. So welcome everybody to our first physical sciences and machine learning Nexus talk of this year. We have these usually roughly every other Tuesday during the quarter. And our first speaker today is Ryan McCarthy. So a homegrown speaker, currently postdoc at the chemistry department. Ryan has a very diverse background in many different things. He has worked on, well, his PhD and masters are from Stanford in geology. And there he worked on solid state NMR, doing experiments on dopants in solids and spin glasses. After that, he was for a little while an engineer at Caltech, developing methods in new inorganic polymers. And then he came here where he was a UC president's postdoc fellow, in which he sort of developed overall this convergent method for problem solving, combining both sort of experimental and theoretical inputs using machine learning. And what's particularly pleasant in this case is that he brings the average age of our speakers down by about 10 years or so. Take it away, Ryan.
Can everybody see my screen? I got a nod from Craig. Great, thank you. So yeah, and the only thing Kieran missed out was the master's. It was in chemical engineering, and I hope you'll see some of this chemical engineering or engineering sort of framework in this. And my talk today is going to be about learning convergence, more or less reverse engineering electronic structure, whether that's observed experimentally or calculated with some method, and to use this to solve multidisciplinary problems. And I think I'll start with the sort of first part up there on the learning convergence. And I think if you asked someone in high school, they'd probably give you this viewpoint of knowledge that's got these pillars of, basically what we have as departments. But I think as you go into college and as you ascend through academia, you learn that those pillars are a little blurred. The lines between them are not so clear. And on top of that, there's vast chasms between these pillars of sort of insight or problems that are unanswered, as well as large space above these pillars. And I think it's these big chasms that NSF has really been trying to get at with one of their 10 big ideas, the one that's called growing convergence research. And I will say it's not the first time we've tried this. It has many different names: multidisciplinary, interdisciplinary, convergence. It's all more or less the same thing of trying to bring together lots of different fields. And I would argue on this one that just like if you take any old professor and you put them into a math building, they're not suddenly going to become a mathematician overnight. Convergent science is the same way. It's a unique set of skills and tools and a lot of sort of cross-language thinking. And so if you just put a convergent scientist in a convergent science building, they're not necessarily going to be a convergent scientist. But on that note, I think there's a large advantage to become a convergent scientist and to use a multidisciplinary skill set, because it gives you a lot of great ways to think. But at the same time, it does cost a lot of time to learn that and a little bit of your belonging and who you belong or who you fit with well. And you don't have to take my word for this. There's a long series of literature actually studying this problem of sort of convergent science. And I think the best paper of the last year is probably this one I've got up on the board, the diversity innovation paradox, which really shows that innovators in their field, many of which in the paper they use examples of household names, take a deficit to their career when they try and mix fields together. And part of that's because the challenge of mixing fields, but also then sort of you're lost in those chasms. But on that note, I think that machine learning presents a really advantageous method to sort of converge a lot of different people together. And part of that's because machine learning has a lot of ways that you can learn machine learning. If you're any of the physical scientists, you usually have a pretty strong math background. And so it's an easy direction, whether you know statistics or calculus, to start learning machine learning. And so in this talk today, I'm going to use sort of machine learning as the glue that holds everything together. And I'm also going to use the lens of electronic structure, as I mentioned in the beginning. And so for the first sort of 2/3 of my talk, I'm going to talk about detecting chemical weapons, chemical warfare agents. And like all machine learning, you usually have some data and you have some model that you train or learn. And so the data I'm going to be using is a really neat type of spectroscopy called J-coupled NMR spectroscopy. And we'll be using both computational and experimental versions of this. And the models that we're going to train are blind source separation methods, a large class of machine learning algorithms. In the last third, I'm going to dive into some of what I've been doing over the last 2 years, which is improving density functional theory. And in this, I'm going to use density functional theory predictions. And I'm going to use hierarchical density-based clustering algorithm for the machine learning part. And I'm going to use it to actually, instead of trying to predict new molecules or things that are unknown, I'm going to use it to sort of work backwards and study how density functional theory is coming up with its predictions and what things are similar or different from each other. So let's begin on the chemical warfare. These are chemicals that are used as weapons because of their toxicity. And we have a lot wrong in the world today, and so I don't really think I need to give examples of this. And I will warn you, if you do go look up some examples, be a bit ready to be disappointed in humanity. And it's because of how egregious these weapons are that the OPCW, which is the Organization for the Prohibition of Chemical Weapons, came together. This includes all countries in the world with the exception of 4 that are not quite full members yet, but most of which have expressed interest in becoming full members. And in particular today, I'm going to be looking at the Schedule 1 toxic chemicals. So these are the worst toxic chemicals out there. And I'm going to be looking at the organophosphates. And for the sort of large class, these are basically include a phosphorus atom, sort of in the center of the molecule. That phosphorus atom is bonded to an oxygen, and then it has these little groups around it that I'm going to call R groups, where you can swap in all sorts of different types of molecules that might exist on their own. And this problem or question here is really a problem of chemical structure, and that's because these organophosphates have enormous flexibility. There are millions of different organophosphates that you could create. And that wasn't first realized by the OPCW. They started out just making a database of each individual chemical, thinking that if they could collect the spectra on all these, we'd have a way to diagnose them. However, there are over 1 million different types. And on top of that, you can't just say any organic phosphate is a weapon. There's some that are used as pesticides, very important for crop yields in developing countries. And there's also a lot of organophosphate type materials or very similar structured things that have the same composition that are fertilizers. And I have some examples of those molecules up there. There's also problems in chemistry's structure. And this is, of course, the OPCW, funded by the nations all over the world, has a beautiful analytics laboratory headquartered in the Netherlands. But the equipment, funding, education, and consumables are a bit limited. They're not spread all over. They're just located in the Netherlands. And so if you were part of this lower picture there, that's part of the Indian peacekeeping force doing some UN border assistance on behalf of the Democratic Republic of the Congo. They don't have access to any tool that they can use to look for these, whether they suspect them or not. And so what's really needed is an inexpensive chemical analysis tool that can discriminate the structure. It's got to be portable. It's got to reach conclusions on its own, and it needs to not depend on a lookup table. And so this is actually where I first got introduced to the problem. Up there on the board, I have one of my colleagues. This is Derek Caseman, who's a staff scientist at Los Alamos National Laboratory and first reached out to me over a year ago. And he had been developing a new type of spectroscopy using the Earth's magnetic field. So this is a nuclear magnetic resonance. It's looking at the nucleus and how it responds to the nearby electronic structure. And while that instrument he's got there, very homegrown looking, is pretty big, that's the old one. I don't have pictures of the newer one. The newer one is much smaller and can operate on sort of milligrams of, or milliliters of material and get pretty good spectra. This is also using the Earth's magnetic field with just a smaller little magnet to up it a tiny amount. And so this is incredibly low. I have up there some of the measurements in Tesla of instruments at UCI, 18 Tesla, and the Earth's magnetic field is far, far below that. And it's basically everywhere on Earth. Now, there's some interesting physics here, though. And let's consider, if we're just trying to understand the complexity of this problem, two different types of nuclear spins. We'll have nuclear spin S and nuclear spin I. And we're going to say there's 3 symmetry equivalent sites that have whatever atom is making up nuclear spin I. The electronic density or electronic structure around those nuclei will control the electronic current density, which controls the Zeeman splitting, which is what you'd see if you were at very high magnetic fields. So it's at super duper high magnetic fields up there. You just see the Zeeman splitting. It also controls or mediates the hyperfine interaction, which results in a indirect dipole coupling, which organic chemists would probably know as the J coupling, or NMR spectroscopists. And that's what you'd see at no magnetic field. You just see the effects of that indirect dipole coupling. Now, let's put up on the board something that most of the chemists in the room are probably familiar with, high field NMR. This you can calculate using a first order perturbation theory, and you see that nice J splitting. So an organic chemist student can usually figure out sort of some rough structure or abundance of different spins based on the J splitting and based on maybe their chemical shifts. Now let's take a look at low field NMR. This is that same system in low field NMR. We get over 16 peaks. It requires an exact theory or pretty close to exact theory in order to calculate this. And it contains the maximum of information you can get from nuclear magnetic resonance. So it has effects of the J coupling and effects of the chemical shift. And you may also notice there's not that nice little J spacings in there too. So a lot of the tricks that we'd use normally don't apply. It's more or less information overload. And as an example of this, I have some of these organophosphates at low field up on the board. And you can even see weird things. So there's some phosphorus shifts that are far above the proton shift there on that far left. And that's a result of a humongous J splitting, things that you would never see in a high field NMR. And so here exists a more or less grand task. There's an entirely new spectroscopy technique. This not entirely new, but pretty new and pretty different. In total, there's about 30 published spectra that have originated from this low field NMR. And there's only a few home-built spectrometers worldwide that can do this, even though they're relatively cheap to make. And so a grant that actually just funded, and in fact, the funding actually just started yesterday, was one that I put in with Derek Caseman at Los Alamos, where he's the PI on that. And his part was really coming up with some instrument development to make his current instrument even better. And a big portion that I contributed was trying to use machine learning and some different analysis techniques to help it go from a complete spectrum at the start all the way to a chemical structure at the end, so that you can identify these. And so what I'm going to cover today in this section is some of the preliminary work that went into that grant. In the biggest picture, what I did was constructed a more or less decision tree. Right now, no machine learning in this decision tree, but that is planned for the future. You'd imagine taking some unknown sample and collecting a spectra on it. That spectra itself is already quantitative, so you can measure the different peak areas and account for the magnetic receptivity of each nuclei, and you can get out the composition. From there, you can determine if that has a possible composition closely related to organophosphates. Derek was initially opposed to having any sort of matching table, but a matching table would save a lot of computational space. If you have any peaks that match up with known chemical weapons, of which there are only a few that have been synthesized and studied in depth, you can right away know that it's probably a possible weapon. But if you don't have that, you would then need to analyze the spectra and try and determine its structure. So that's what I'll be diving into a little bit more. And of course, if that structure contains weapon-like groups, you can assume that this is probably a chemical weapon. And so on this note, as I mentioned before, those organophosphates have those different R groups around them. And the thought was, can I use blind source separation to separate out the parts of the spectra that relate to these different R groups? And so up on the board, I have a number of those chemical weapons. And you can see in some cases, those little peaks might match up with the peaks in the chemical right below it. And that's possibly because they're sharing a very similar R group. In this one, most of them have the same R3 group, for example. So what is blind source separation? Essentially, it splits complex data into smaller chunks. It actually amusingly is maybe you could call it some of the start of machine learning. It comes from a field called chemometrics, which has basically died out by this point in time, but was started in about the 1970s and contained the very early statistical chemistry and statistical chemistry approaches on spectra and different compounds. Also, these are known by many different names. You can find all of them by looking at blind source separation and reading the Wikipedia, but do keep in mind that there's a lot of different flavors and it's used by a lot of different fields. Essentially what we do in these is we do a matrix decomposition or factorization. And I usually like to think about this as something that most people can approach. So how about bread? All our bread's made with some relatively simple ingredients or components that I've got up there in yellow. The recipe would tell you the different amounts that you would mix those together. And in the end, you'd end up with some bread. And that bread is more or less the observable matrix, or this would be all of our spectra that we can observe. And what the machine learning is going to try and do is split it apart into these are the different components or different chemical species, what their spectra would look like on their own. And these are the different amounts that they are added together to make up that observable. And if we take a look at maybe like a flow diagram of what this looks like, in reality, there's all these different chemical components or different chemical features, and they come together to make that observable in the green there. And what we're doing is really working backwards, and we're using a loss function to do this. Our loss function is, of course, trying to get that yellow block there, an S, the little components and the weights of those components to match up with the observable. And it also includes some extra parameters in there. So it's probably using the variance or some correlation, possibly arguments of pixel purity or other statistical or mathematical requirements. And it's trying to satisfy and minimize error or some sort of function on all of those to maximize the reproduced M matrix. And so as a concrete example, just to highlight again some of my own work as well, and some of the stuff I started doing back in 2015 on this, let's take a quick look at cements. Cements are used worldwide, very important for people. We've probably all seen it. The key to cement is a mineral called B-lite that holds the cement together as it's in its unwet form. And once it gets wet, this decomposes and results in the long-term strength that you get in cement. B-lite, though, is not stable at room pressures and temperatures where we also often need to mix and make cement. And so what you actually do is you take a chalco-olivine, or a different phase of that material, and you add in a tiny bit of aluminum at high temperatures. And this makes that B-lite. And for a long time, we didn't know where even in the chemical structure, in the crystal structure of this B-lite, that aluminum went. And part of that problem is because when you're trying to make these samples, you're trying to look at part per millions amount of aluminum inside a crystal structure. And you only need a very, very minimal impurity, like a luminosilicate glass that has a large amount of aluminum, to basically wipe out and confuse your NMR spectra such that you can't interpret it. So it's not the first time NMR had been used to look at this, but the conclusions had always been mixed and a lot of arguments ensued. In this case, I took a lot of different collection of different samples of this B-lite, all of them having a little bit of aluminum added in it. And I used a blind source separation tool called Simple ISMA to split apart the different components based on the variation in all those different samples. And in this case, I got 4 different components. 2 of those were very easy to cross off as not relating to the B-lite, and that's because one was an exact match for luminosilicate glass, and another was actually the rotor inside the NMR that you need in order to contain your sample. So we knew what that one was. Those are components 3 and 4. And then for component 2 and 1, we still were unsure which of those was related to impurity phase or related to the bulk B-lite that we were trying to look at. And in this case, we used another technique. And I think that's really the key to using machine learning in the physical sciences, is it's one of many different approaches that you use to solve a problem. Just like you might use DFT and some spectroscopy. In this case, we're using some spectroscopy and machine learning, and could even have used DFT or something like that here. Anyways, when you look at the different concentrations, you get an almost exact match for component 1 being originating from the very trace amounts inside of the bulk system. And this led us to a very exciting result for the cement community, but basically told us that the aluminum had to be in a system where it was tetrahedrally coordinated by oxygen, which in the structure of B-Light means that it can be in only one site, which is the silicon site. And so then we could begin to think about maybe the thermodynamics behind that phase stability and where that originated from. One thing that did bother me, though, in a lot of the blind source separation work that had been done in the literature that I had read myself, was we more or less picked techniques based on what was convenient or what might give us the best answer or what we thought might give us the best answer. And it's very hard to know when you're in a really unknown setting. You don't necessarily know what the end product is. And so one of the things I did and got out just last year is a large benchmark on a bunch of different blind source separation methods. And I did a benchmark on synthetic spectra, where I actually created very much NMR-like spectra. And I combined those together, so I more or less behaved like the physical system. And then I used these blind source separation techniques, and I benchmarked them all against themselves. A key thing for what I'll talk about later is that in this work, I came up with some rather cool algorithms that use a combinatorial approach to match up all the different original things from the predicted spectra. Because there's no guarantee that your first input component that you might have mixed into something is going to be the first one to come out of your blind source separation technique, or even the same first one to come out of that blind source separation technique if you use it again and change a few parameters. The big takeaway of this, though, is if you're thinking of using some blind source separation methods, I've done a lot of work and sort of picked out the top performers out of all the rest of them. And so you don't need to guess maybe which one's going to work best or try them all out yourself. And then I'll use, actually, in this later talk, some of those same top performing techniques as well. And up on the board, I have just an example of all these different techniques done over about 7,000 different spectral arrangements. And you can see that there's a wide range of algorithm performance, even on a given algorithm, sometimes can perform really well or very poorly. And so if you use sort of a statistical approach and you want the very best that will happen most of the time, some of those are like the fast ICA method, non-negative matrix factorization, or simple ISMA. Okay, let's return to those chemical weapons. For this project, we used a bunch of synthetic data that we made up using a spin dynamic simulation. So this is kind of like density functional theory. If I know a number of people in the audience have used this, but instead being applied to the nuclei. It's, of course, a quantum mini-body problem, and these nuclear spins do have a somewhat significant amount of spin correlation amongst themselves. But luckily for very long or large systems, it seems to be only a couple, 5 or so bonds away. So you can get away with usually doing sort of applying a basis set as opposed to that exact theory, and simplify the problem quite a bit. We still need, for that spin dynamics calculation, you do need some guess of what the J couplings are ahead of time. It's not electronic structure, it's just a nuclear spin calculation. And so that you can get from high field, though do keep in mind, if you're a chemist, high field J couplings usually don't have the sign on them. And so in that, we usually converge with density functional theory. While it usually didn't get the J couplings as accurate, it did tell us the sign pretty accurately. And if you ever want to do any machine learning on your own, I highly recommend checking out the, on density functional theory calculations, checking out the quantum machine data sets, because there's a lot of data there that you can test on and practice before needing to do any calculations or spend a lot of time on your own. For these molecules, one of the R groups, we're going to put a fluorine, and this is the Schedule 1A if you're the OPCW. So they're the most top of the list, most toxic chemicals. For the R2, we're going to have 4 different options. Most of them are alcohols. And for the R3, we're going to have some of those same options and then a few additional ones. Go into more detail in that second. And first big takeaway was that you can use non-negative matrix factorization on some of these synthetic or real data sets. You can split apart a bunch of different peaks. And just an example of this, I have the proton region of these low field NMR spectra there. And you can see a bunch of different, very much NMR J-coupled splitting spectra up on the board. If we zoom in on some of those areas, the most important part is that you may see there's some overlapping peaks. These peaks are overlapping with each other. And the non-negative matrix factorization, even with rather small data sets, does an excellent job of splitting apart peaks that are overlapped on top of each other based on their different variations. So if you have overlapping peaks, normally as a chemist, modeling those all out, or any peaks really, that applies to a lot of fields, it can be difficult to do, but non-negative matrix factorization does a good job of splitting that apart. All right, let's take a more in-depth look now. So on the board right now, I've got some graphs. On the left one in green is the experimental data. Or no, this is synthetic data. We'll get to experimental in just a moment. And each of these different synthetic data have different components in them. So this top one's got an ethyl phosphorofluoridate. And some of those others have methyls, pentacholeuls, isopropyl groups. On the right-hand side in purple, I have the known R group. So this is what that chemical is known to have on it. And what you might guess or might see right away is that on the left-hand side in green, we definitely get some of those same big blocks. So those same related points in there that is showing that component 1 or component 2 or component 3 matches up and is found in rather high abundance in those samples that are listed there on the left-hand side. And so what's rather cool about this is you can take a look, you see something like that. Try and circle with my mouse on the board. These isopropyl R groups on isopropyl and R3 match up very well with this isopropyl known to be in the R group. Something like this penacholeuel, we can start to see some of the limitations of this approach in which the penacholeuel 1 and the penacholeuel 2 both have basically the same chemical signature. So you can't tell the penacholeuls directly apart, but you can tell that both of these have penacholeuel in them. You can also see, and it's a little scattered right now, but all these different boxes that sort of have this slanted or sloping looking appearance to them. And if you were to look at each individual one, you could match those up. So this group right here should have an ethyl in the R2. And in fact, it does have an ethyl in the R2. This one right here has an ethyl in the R2. Again, has an ethyl in the R2. There are some limitations though. If you take a look at the methyl groups, so all the ones that start with methyl, you'll see they don't have a really clear match in any one given thing. Components 8 and 9 do match up somewhat with that ethyl group, but it's not an exact match every time. And in fact, it splits that methyl group into actually 2 different components. This is, of course, because the spectra that are coming off of having that methyl group vary widely. And that's because the J couplings of those in that local electronic structure changes quite a bit from one molecule or another molecule. It has a very strong coupling to the neighboring groups. What we can do, though, is with just this input data and knowing that there's these different things that should be in that sample, and then finding that there are some different components that appear to correlate well, we can use Pearson's correlation coefficient and run through each of these and try and match up each of the R2 groups and R3 groups with some different chemical. I know the font's small up there, but you can see that basically 1, it ascribes to being the penacholeuel 1. That second component, it ascribes to being the isopropyl group, and so on and so on. And the really neat thing about this is that you can start dropping out members of the data set, redo your machine learning on it with some of those as unknown, redo your Pearson's correlation assignment. So you're assigning all these different chemicals just based on the ones where you did know. And it usually does a very good job. And so on the simulated data, you get about 75% accuracy for both R groups, where it can correctly assign them without having an exact match, just knowing that there are other chemicals that had the same R group in them. On that same note, if you look at just the R2 groups, it seems to do a very good job matching those up. Basically, on the synthetic set, we can get 100% accuracy, matching an R2 group up to another R2 group. If you look at the real data, the accuracy does go down a little bit, and that's because these spectra can become very noisy, and we're also working with a little bit less data. Also, we have a little bit fewer choices to choose, and that's because there's staff people that make up all the molecules for Derek at Los Alamos, and we don't really want to be making up some of these toxic chemicals, so we make up some analog ones instead that are still organophosphates, and we use them to take a look at this. What this basically shows, though, is that you could imagine throwing in some unknown chemical and having a database of these different R groups with other similar organophosphates, picking out organophosphate groups that you think are close to this unknown chemical that you put in, and then doing a matching. So this is not quite the lookup table where you need every exact chemical, but it's a lookup table of finding the same R groups based on their structure or related structures. And so this was, I think, probably the key thing that really helped that machine learning section there. On training these models, though, there are a lot of different settings that you can set, hyperparameters. There's a lot of different initiation states that you could start with. So whether you use a random initialization and you try and converge something, or you use one of the techniques that starts out right away on some hyperparameters, or you use a singular value decomposition as your starting matrix, and then you try and optimize that. Each of these varies for the different blind source separation techniques that you might use. And of course, you also have to select one of those blind source separation techniques. So while we'd want to imagine that we have an easy slope that we can converge down to come up with our final components, it's not that simple. Usually you might imagine it's actually a very rumply surface. And you could also then envision that you get stuck in some local minima. And so on that note, actually one of our previous speakers at the Machine Learning Nexus, Klaus Robert Mueller, pointed me towards one of his papers when I had expressed some frustration with this. And this is one where they looked at a lot, basically dropped out sets of the data and kept running PCA to determine how stable their outcome was. Also, this is some other ideas that are very similar to this is like the random forest simplified model, random forest model. We have lots of different decision trees, and you more or less pick the outcome that was most voted for. And so this sort of concept gave me the idea of a blind source separation model voting. So where perhaps you could run many different models, have them all vote, and take whichever votes, sort of concepts had the most votes for them. And so in this regard, I came up with a way to do this, actually using that matching approach that I had used originally in the benchmark, but instead of matching to the real answer, just having techniques match to the answers of other techniques. And what I often did was ran the non-negative matrix factorization, simple ISMA, and fast ICA, some of those fast, best performing techniques. And what I found by doing this is that you can then get much higher quality predicted components. So before, where you might have a lot of different issues of noise if you use any one given blind source separation technique, by sort of adding them all together, you might imagine that they try their best to converge to the very best answer, but they might sometimes get stuck in local minima. And you also might guess that all these techniques are more or less designed by different people to do the exact same task, come up with that exact real answer. And so you all try or do pretty good at getting down somewhat close to the real answer. And then it just really becomes a question of whether you group things together, whether you bin one component that was predicted to be similar to another one and say they're the same and start averaging them, or you split those apart and you say they're competing ideas. But what this did was really allowed and can be used for is predicted components can be substantially less noisier than any one given technique. And you get a lot less cross-mixing, where a component from one sometimes gets mixed into a component from the other. By doing all this model voting, you more or less help split everything apart into their own given areas. Another fun thing that I crossed upon while taking a pretty good look at all the different data sets that Derek had sent along my way was the effects of relaxation in the J-coupled spectra. There have been some discussion that you might be able to see some effects of relaxation in the J-coupled spectra, or if more or less the strength of magnetic field is so low that all that will really get washed out. And while this is a rather exciting finding, I will say it's so exciting that we're retaking all the data again, just to double check and make sure that we're actually seeing what we think we're seeing. But what we did here is we took a bunch of different J-coupled spectra at different T1 relaxations. And I was actually doing this to try and model the noise a little bit better. But what I found was consistently, the blind source separation techniques would split apart two different sets of data. And when I showed this to Derek, Derek was like, oh my gosh, this is what I had thought might be in there. There's actually two different R groups in this particular molecule that we were looking at. And one of those R groups might have been thought to relax very fast because it had access to an oxygen atom nearby. Those are the ones in blue. And the other R group was thought maybe it relaxes a little slower, though finding some evidence for this was pretty difficult. And what the blind source separation did, though, is at each increasing T1 relaxation, we suddenly see a rise in this green spectra here, in the amount of this green spectra being used to model the data, and a decrease in the blue. And that's because that blue is relaxing so fast and is basically being all done. You've got all that intensity, where the green is taking a little while before it's fully relaxed to be a larger part of that data set. Some quick summary and next steps before I move on. But more or less, this gave the proof of concept that we needed. And it was quite exciting that that grant got funded. I'll probably be spending a lot of the next couple months working on that. But the key thing is that there's a correlated assignment of different R groups is more or less a success. And the components are detailed enough that you can analyze them individually. So sort of more or less a next step beyond this. But if you could imagine separating out these different R groups, you could then imagine maybe counting peaks and trying to analyze in much greater detail what that J coupling might be and how that relates to the structure visible. So next steps, though, is automate a lot of this so that it can be done very easily and come together without too much input from the chemist. But I will like to mention in that broad diagram I had, in most of these cases, these diagnostic tools will be used to come to a decision whether to hold this sample and send it on to a real chemist to take a look at, because I don't ever think machine learning can completely replace the chemist, or to know that this chemical or sample is not at all related to a weapon. Some other future steps is working on more of the R groups, putting that model voting together so it can work with all those different R groups, and putting this whole thing into a very large machine learning driven decision tree. I will pause at the end for some questions, but just looking at time, I'm going to keep moving on. So for the second part, I'm going to talk about improving density functional theory. And density functional theory, as most people in the chemical driven sciences, whether that's looking at things in the atmosphere with a more earth science perspective, or minerals, or condensed states and solid state systems, density functional theory is often our go-to tool for computing the electronic structure of something without knowing really fine details and without spending an enormous amount of computational time. And the real key to density functional theory is you try and come up with some density. And from that density, you get all the properties that you might ever want. That includes wave function, nuclear positions, energy, current density if you're doing things like NMR. But the real key to coming up with that density, at least in the practical computational sense, is coming up with a non-interacting single particle potential. And there's a lot known about these potentials and how you might put these things together. And a lot of things are very universal about those potentials. But there's one part, which I think anybody who's ever run one of these density functional theories probably knows, called the electron exchange correlation functional. That is really an approximation. And it's not something that we know exactly. And so therefore, it's the source of all the mistakes or errors in density functional theory. However, when we first got some of these early density functionals, exchange correlation functionals that worked well, this was the key thing that really enabled density functional theory to go off to the races and enabled people worldwide, people who had never used density functional theory before, to then start picking up these softwares and applying them to real chemical systems. And just as some examples, you might have heard of some of these: PBE, B3LIP, HSE, TPSS. They go on and on and on. And part of that's because they are approximations. And we don't know what is the best approximation out there until we more or less come up with one and then try and test it. And because of this, there are over 500 different exchange correlation functionals. If you get access to some of the libraries out there, you can have those 500 right at your fingertips. And then, of course, you can start playing games that probably contributes to global warming, going through and testing all these different 500 exchange correlation functionals. And there are so many functionals because there's not necessarily a straightforward path or method to improve these density functionals. we have some good guesses, but a lot of it takes some really hard theory work, piecing together things and always comparing with higher level or more expensive levels of theory to get a better idea of how that theory might work together. Now, in these exchange correlation functionals, part of why there's so many is that there is definitely a religious divide among computational chemists who come up with these. And I would say the divide is among the exact crowd and the empirical crowd, where the exact crowd is trying to come up with really the physical underpinnings, first principles approach to what should go into these exchange correlation approximations. Whereas the empirical crowd is saying, I don't care about the physics as much. I really want it to work exceptionally well on a different class of molecules or solids that I'm interested in. And so they go through and they try and come up with something that matches really well to that, or they think really hard and scratch their brains and come up with some better theory that describes the exact conditions. And of course, these are just n members of the entire spectra. You can imagine people in the middle mixing together some exact attributes with some empirically derived ones and trying to come up with better and better tools. However, on this note, and as pointed out by this diagram that I have up there from this paper that came out in 2017, as time went on, we kept getting better and better densities produced by these exchange correlation functionals. And then all of a sudden, in about the 2000s or so, the densities started getting worse and worse and worse, even though something that this does not show, the energies and errors of some of these later functionals began to increase and increase and increase. And part of that's because these exchange correlation functionals are optimized on energy. They're not optimized on the densities. And you could imagine for any given system, you slowly work your way down with a process called self-consistent field, where you start at some guess of what the electronic density is. You improve it a little bit, you calculate its energy, you improve it again. And this is because one of the fundamental theorems says that if you get the lowest energy, you have a density that's in its ground state for that given method. However, you can begin to see that there could be some problems here. If you have a density from one technique, and it's the same as a density from another technique, and it's the same as a density from another one of these exchange correlation functionals, you could all have the exact same density. But that doesn't necessarily mean that they're giving you the same energy, because the functional is used for both optimizing the density and calculating the energy. In the same regard, you could imagine that you could have a large number of densities predicted by different functionals, even though they all get the exact or correct energy. So they've got differences in densities, which means that some of them must be wrong, even though you've got the right energy. And so with that, or no, not with that, but the stuff I'll talk about, I'll give a much more in-depth discussion at APS. And I'd also like to point out that this is with some more theoretical contributions from Stefan Vukovic, who was here at UCI for a while as a postdoc. And also at UCI was John Kolowski, a graduate student in Kieron's group, and some of Kieron's computers, and Insight was also used in this project. But let's take a look at a technique called density-corrected DFT, which we're going to use to analyze all these different density functionals. Okay, so we've got a density from one method and a density from another. What if we take that density and we evaluate it using one of the other exchange correlation functionals? This is essentially the thought of density-corrected DFT. So you evaluate one density using another functional that wasn't used to make that density.
Sorry, Ryan, you have about 5 minutes.
5 minutes. But what we realized is that you could actually start doing this again and again and again. And you could use both that density and energy calculations from A on B and B on A to start building a matrix. And I thought I had a little more time, but I must have lost track of that. So I'll speed up here, but you could imagine that for any given functional, evaluate another density. If you're very similar in density, you'd have a very similar energy. Whereas if you're very different, you'd have a very different energy arising from those. And you could do this in multiple dimensions. So again, if they're very similar, they'd cluster at the origin of all these different dimensions. And if they're very different, they'd begin to cluster away from those origins. And to do this, while you could look at one individual molecule and run this calculation using all these different functionals, A, B, and C, what we did was use a whole bunch of different molecules to get a really big average picture of what the system was going on. And we averaged all these different molecules together, and we did this on a whole bunch of different functionals. So functional after functional, we used over 32 different functionals, and we ended up with a giant data cube, basically, of all these functionals evaluated on lots of different molecules and evaluated on each other densities, so the different densities that different functionals would make. We then used a technique that I found called HDB scan. This is a hierarchical version of the DB scan, which clusters based on density. So it tries to look for a little sphere around it where it has the same number of points. It then tries to link all those different spheres together. And so quickly show a nice picture here. You could imagine all these different points are all related. The ones in darker purple are closer to each other, and the ones in lighter color are further away. And from with that, you can construct a large tree. From that tree, first thing I did was tested to make sure it gives rather consistent results. The big takeaway from this is that it does. But then looking at what results it told us, or what it showed, up on the board, I've got a lot of different density functionals. If you zoom out and look at them in the biggest case, so you minimize the amount of clustering that the algorithm will do, you see that nobody is related to each other. And that makes some sense. As you start changing that parameter, eventually working towards everybody being in one big group, you start to see some groups that are rather curious. One of them I call the old school group. So this is some of the very early density functionals came up. This is like LDA or GGA type functionals. You then also see another group that's more or less the hybrid functionals and some of their friends and a bunch of outliers. This is a 2D projection, by the way, of this 32-dimensional space. So it loses some things, but follow the colors are a little more accurate or descriptive. You then can keep upping that number and look at even bigger clusters. So we suddenly see it, the old school and the hybrid and friends lumped together as being more similar to each other. And then some other functionals. These ones are more or less the modern go-to functionals that are exceptionally good at small molecules, such as the MO6 or omega B97X. And you also see some functionals that are way out there. These are exceptionally different. And what is rather interesting about this is these exceptionally different functionals are ones that were optimized using linear regression, so using machine learning type methods. And while they give exceptionally good results, they come up with densities that are very obscure or very different from everything else. Moving along, in the biggest picture, they're all density functionals, so they all cluster together. One last fun thing we did was took a look using the same technique, but looking back at the molecules. And a cool takeaway from this in just a few seconds is you can see that there's lots of different groups up here. These groups relate to the different type of bonding in these molecules and the number of electrons inside these molecules. And you see that they have all different distributions. But another takeaway you might see is that there's a lot of things that cluster together. And these are data sets made for benchmarking that people came up with. They have a lot of redundancy in them in some areas and a lot of missing space in others. And so using this technique, you could almost imagine coming up with better benchmark data sets that explore more of the space that you might want to optimize a functional if you are doing something with linear regression. And here's real quick what that looks like if you were to spin this cube, adding a third dimension. Again, you see some of those orange things up in the top are fluorides. They're very different than all those things lumped together basically are carbon or hydrogen bonding dominated chemicals. And what's also really cool is some of these chemicals that lump together in this picture, which is how similar functionals perform on them, have very different energies. But when you look at them in this space, you learn that they're actually, the functionals at least, are performing very similar on those. Okay, so in a quick conclusion, hopefully I showed that blind source separation is a useful tool for simplifying spectra and signals. I have lots of code on this stuff that I'd love to share, or GitHub repositories that I'd love to point you towards if you'd like to know more. On the clustering end of machine learning, I hopefully showed that it can provide some rather unique insight, even on data that normally people might not think to take a look at, like that density corrected DFT comparison. And overall, machine learning sort of provides some maybe bridges between different disciplines in STEM. Quick acknowledgements. Thanks to Kieran Burke Research Group and National Science Foundation grant that funded me for a little bit, as well as my collaborator, Derek Caseman from Los Alamos National Lab and that project. And then, of course, the UC President's Postdoctoral Fellowship. If there are any graduate students in the room, I hope that you take a look at this fellowship. It is an awesome opportunity for anybody interested in a faculty career because it provides a hiring startup incentive for anybody who has one of these fellowships. It also has a rather good mission of trying to diversify academia. And a rather fun fact about it is it doubles your chances of being hired into a faculty position outside of the UCs, just having one of these postdoc fellowships because it's rather prestigious. So with that, if there's any questions, I'd love to take those now. I can stay on and chat or set up chats later in the week if we don't have time to get to it now. And my email's up there for that reason.
Thank you very much, Ryan. I see there's a question from Luke. Do you want to ask it yourself, Luke?
Yes, I could if you can hear me.
Yes, we can.
Cool. So I would first like to ask, what do you mean by friends of hybrid DFT functionals? Like, what is this friends category? They are not hybrids?
They are, most of them are hybrid functionals. There's a few that get lumped in there because they're sort of on the edge of GGAs. They're GGAs, but they're more recent GGAs, ones made in the last couple of years, like last 20 years, I guess.
I see. Yeah, thanks. I think the more interesting question for this audience would be, you mentioned how machine learning is a good tool for converging different scientific fields. And I'm wondering how it relates, in your opinion, to the abstraction in machine learning, that we are trying to abstract a layer. In this description, you start with chemistry and you then try to link it to matrices to relating properties, that sort of abstraction.
I think it really relates in many ways to in like a larger picture. This might be too broad, Unambi, but with the specifics of your question, but relates to the scientific method and how we do science. We use data and we try and learn some conclusion, and we do that all the time. The only thing different about machine learning is you add in some mathematical framework that limits the conclusions you can come to. And even in machine learning, we see a lot of times that you'll use one method, you test it out. If it doesn't work, you try another one, you try another one, you try another one. And if you talk to some of the folks in computer science also to how much time is spent just trying to optimize models, which is a lot of what we do in the sciences. We spend forever trying to come up with a better description of bonding or a better way of understanding how galaxies spin. And so it's not too different.
I see we have quite a few more questions. Assuming that the machine has put them in order. The next one is Professor Rachel Martin.
Hello. So I enjoyed your talk, Ryan. So I know it's supposed to be the machine learning seminar, but I can't resist. Can you tell me about J-coupling and relaxation in those J-coupled spectra? That sounds like a really cool result.
It is, and that's why I told Derek, we gotta retake this data and we gotta find probably even another molecule that shows that same thing. But basically what we're seeing is that the R group that has, I believe it's a little methyl group that has an extra oxygen between those two, relaxes just a little bit faster than the group that does not have the oxygen between the methyl group and phosphorus. And this is very noisy data, so that'd be another caveat that I'd say, but pretty consistently and across simple ISMA, ICA, and the non-negative matrix factorization, you see one peak and then you see one slightly lower shifted peak. So it has a slightly lower frequency, which is roughly what we'd guess based on the J coupling and then the frequency known at high field.
So is your collaborator synthesizing the molecules? Like, can you label it?
We could.
Like
He's done everything I've worked with him on is like natural abundance, non-labeled stuff. But
if you stuck an 017 in there, then that
you'd
would
know
change it
for
a lot.
sure.
Yeah, you would definitely know.
And I don't know. He actually doesn't actually have to make any of them all, or he's not allowed to make. Trained synthetic chemist, and then in the labs he's not allowed to make any molecules. So, but that is a great idea.
Well, it's really interesting. I'm looking forward to seeing how that comes out. Yes.
Okay, next was Timothy. Are you there, Timothy?
Yes, I'm here. Had to click a button. So I understand that your DFT, density similarity plot, I'm trying to find the right words for it, tells you that the newer functionals are giving densities that are really, really weird or very different from older ones. Does it just kind of tell you which ones are similar, or does it also like kind of point you in a direction of how, maybe what they're doing wrong to get weird densities so that you could hopefully get a better density and also be getting better results? Does it give you information in that direction?
The hope would be the latter, that it would tell us some direction to go. What's very interesting about the density similarity, it's not a measure of accuracy. So we don't actually see whether these functionals are more accurate or less accurate. But that's also its advantage. It's not everybody who looks at these looks at the accuracy and how accurate they are. So this is now a new approach, another way to take a look at that. You did mention newer functionals. I wouldn't say they're newer functionals. There's some very cool new functionals that are also up on there that came out of Martin Head Gordon's group at Berkeley, where they are doing some data optimization, so some empiricism, but they're limiting the amount of that empiricism that can be done, so how close it can fit. Whereas the functionals that are probably the weirdest, they actually are known even by their creators to be a little weird. And they tried to make improvements, in even more recent functionals that they made actually plot closer back to sort of home base where the rest of the functionals are. And so they knew that they're probably overfitting the data in their data set, their training set, and such that when you applied them to other things, or even in the densities that they're making, they've got large density errors, but they're getting away with an energy error cancellation. Did that answer?
Yeah, I think it got me kind of in the direction.
Okay, there's somebody called Doug Tobias who was next.
Yeah. Hi, Ryan. Thank you for your talk. Very, very interesting stuff. Quite diverse array of applications. So I wanted to ask if you've given any consideration to, or if you think it might make sense to consider, in your decomposition of the NMR spectra and predictions of the mixtures of these compounds, to augment the NMR data with other, perhaps even more readily available data, such as mass spectrometry, to put some constraints on your predictions.
Mm-hmm. The whole driving goal behind this project and the funding source was something that can be deployed in field. And I don't know about mass specs, or cheap and in field. And I don't know about the pricing of mass specs. I do know they're getting lower and lower in cost.
Yeah, they're routinely employed in large quantities in atmospheric field studies. And Marie could probably tell you more about that.
Yeah, it might be great to know. And on this note, the NMR is it's a Derek Caseman's love, of course, to do this low field measurements. But we aren't pinned to any one tool. And so if there are other techniques like the mass spec that might add an extra key that we're missing, that might make it super simple to split apart whether you're looking at an organophosphate fertilizer versus a pesticide or something like that, that would be very useful.
Right, and also just to, if you've got your R groups right, because if the masses aren't right, then it can't be right? So that it would just be meant as a constraint. Yeah.
And that is not in this project, but some of the, like in the cement project where we're using the wavelength dispersive spectroscopy for concentrations. But there's a lot of different tools and they all blend pretty well together in a machine learning framework.
Okay, next we have Saurabh.
So yes, I have two small questions. Yeah, so so in your first talk about spectra to structure, so your tests, like your training set, like included only spectra from that you generated theoretically using DFT calculations, but your test set included like real spectra. Am I getting this right? And so accuracy like increased.
So you are getting that right. What we first trained on and developed the methods on was the synthetic stuff, but every thing that I showed today is a unsupervised learning technique. So we actually then reuse that same unsupervised technique on the training, on the testing data or the training data. And so it's rebuilding the machine learning model each time. It's a little bit different than some of the other talks that have been previously in this series where they're using something like a neural net, where it trains up a whole system and then applies that system or uses that trained model on something. This one, the goal is to train the model. And so we retrain it each time on whatever data we're looking at.
Okay, and like one more small question. So you use some sort of like a model voting system. So like if a large number of model agreed that this is a particular like alkyl group, then you kind of fix it. Okay. Yeah, so that was like the concept behind it. So like,
yes,
how do you like, what kind of models do you choose, and like how would you give preference? Because I saw like very complex ML models and also simple ones like PCA, and I'm just curious.
So in this case, something like your PCA produces the component, and then you might run PCA pretty much the same way with one little thing change, and you get a second component. And so the matching system is then saying, are these two related? And there's some fun little tricks. First off, where 0 is in predicted components can vary, and how big or tall these are can vary. Because you could imagine, I'm going to use, instead of 1 bag of flour, and I'm going to use one of them, I could use 2 half bags of flour. Those are both results that can come out of the sort of blind source separation approaches. So once you adjust these 2 spectra or 2 different things so that they line up and look as close as they can to each other, then you say, are these the same thing? And that was using just a least squared comparison of across the entire spectra, whether there's a large error between spectra A and B, and using some rather simple things that just say a cutoff of similar by this much or this percent or different by this percent means they're going to be put into the same bin or count as votes for different bins.
So one last question from Dimitri.
Hi, Ryan. Thanks for that great talk. I wanted to ask really quickly, it's actually a related question in the this ensemble approach to the blind source separation. So you have all of these different methods voting. One of the advantages of the random forest method is that each of the trees has a well-defined weight, right? Because combinatorially there can be only so and so many trees. I wonder if you ever thought about an approach where these voting ensembles are composed of models which have a well-defined measure of weight, and if that would be actually useful. It actually kind of applies to the DFT ensemble almost as well. So thank you.
I had not thought about the weighting in depth yet, but that is a really good thought. At the moment, I was just sort of taking the top techniques and then running them each some large number of times with different variables. So maybe a technique is run 20 times, technique B is run 20 times, technique C is run 20 times, with some variable changing in those different techniques. So trying to explore all the solution space with some reasonable amount of runs. But I had not thought maybe we need to wait. For example, we know from sort of the benchmarking approach that some of these methods are very accurate, or not, some of these methods are somewhat accurate most of the time, but a few of the techniques that are usually on average not very accurate can occasionally be exceptionally accurate. And so I wonder if there's some way to wait, if other people match up with this one, this lower grade technique that sometimes is really accurate, but on average is not. Does that mean that answer is closer to the ground truth? That's a really cool thought. No, thank you for that idea. That's got some gears turning.
Okay. Are there any more questions? I don't see any electronic hands. If not, let's thank Ryan again.
Thank you so much, everyone, for your questions as well as coming to my talk.
And I put out a call because I realized this is the easiest way to get the interested people to send me suggestions for speakers that fit in this series. And also, it would be nice to see some more, maybe from math and Earth system science. We seem to get a little dominated by physics and chemistry. I wonder why. Yeah. Okay. Thank you, everybody. And probably see you again in a couple of weeks. Bye-bye.